Hello, Brain Shakers. Welcome to the Brain Shakers Academy. Brave Alistair is here. Now, in today's session, we will be looking at another type of cell division known as meiosis. Now, we have started looking at a series of cell division, and if you haven't seen the video on the cell cycle and the other type of cell division known as mitosis, please do check out for those videos on my YouTube channel, the Brain Shakers Academy, and I will add the links as well to those particular videos so that it's easier for you to get through to them. And when you get to the YouTube channel, do not forget to subscribe if you have not yet subscribed. Now let's get right into it and get an understanding on what meiosis is all about. So meiosis is also known as a reductive cell division, where we have in humans having one parent cell going to give us two daughter cells. And once those two daughter cells have then been formed, they continue with the process of cell division and end up giving us two cells each. That means four cells in total, with each cell containing a haploid number of chromosomes, which is one set of chromosomes within them. Now, the process of meiosis has two major types, or rather phases, where you have meiosis 1 and meiosis 2. Now, let's look at the two varying types of meiosis. Comparatively, meiosis is just like a mitosis in so many ways, and the cell still undergoes the similar stages of the prophase, the metaphase, anaphase, and the telophase. But in meiosis, you find that the cell will have to undergo the first meiotic division before it can go to the second meiotic division. And during the first meiotic division, the resulting number of cells will be two daughter cells. And once the two daughter cells have then being produced, then the two daughter cells enter and complete the second meiotic division, and we end up with four daughter cells. Now, the first meiotic division has the interphase, the karyokinesis, and the cytokinesis. The process of dividing this cell will first go through the interphase, which is the cell growth phase. Then you have karyokinesis, which is the division of or replication of the DNA in the nucleus, and then you end up with cytokinesis. Now, the prophase is considered to be the longest because it has five sub-phases, where you have the leptoten, the zygoten, the pachyten, the diploten, and the diakinesis, and then proceeds to metaphase, anaphase, and telophase, and then cytokinesis. Now, in between meiosis 1 and meiosis 2, the cell does not undergo any DNA replication, and that is why it has a phase known as interkinesis, from karyokinesis 1 to karyokinesis 2, interkinesis. Now, in meiosis 2, the phases are still the same phases as in meiosis 1 and in mitosis. That is prophase 2, metaphase 2, anaphase 2, and the telophase 2. Now, let's look at the first meiotic division and understand what happens in the meiosis 1. Now, in meiosis 1, it is important to notice that it is the separation of the homologous chromosomes. Now, every cell has chromosomes from the paternal and from the maternal side, and those are homologous chromosomes. Now, those homologous chromosomes, they have to then be separated before the cell can complete the second division through meiosis 2, which houses the separation of sister chromatids. So the cell still undergoes the interphase, the prophase, the metaphase, the anaphase, telophase, and the process of cytokinesis as well, to be able to separate the homologous chromosomes before it transitions to meiosis 2. Now, let us look at the individual phases of meiosis 1 in detail. So in prophase 1, which is considered as the longest phase in meiosis 1, prophase 1 is divided into 5 categories or sub-phases. What happens in prophase 1 is similar to what happens in prophase under mitosis. Basically, the nuclear envelope begins to disappear, and you have the appearance as well of the mitotic apparatus and the mitotic spindle fibers. The nuclear envelope here, as you can see, begins to disappear, and you have the centrioles moving to the poles in readiness for these chromosomes that are in here, which are the homologous chromosomes, to be aligned later on as you get to the last stage of the prophase 1. Now, the prophase 1 has phases such as the leptoten, the zygoten, the pachyten, the diploten, and the stage of diakinesis. Now, let's look at those stages in detail and understand what happens to this cell that has entered its first meiotic division and is now in prophase 1. So when the cell has entered the prophase 1, it will have to go through the 5 phases that we have talked about. Now, these phases allow the chromosomes to exchange genetic material and complete the process of genetic recombination. So the first stage is the leptoten stage. This is where the exchange of genetic information begins. So a maternal and a paternal copy of the same chromosomes, each possessing 2 sister chromatids, will be joined at their centromere, and they find each other in the nucleus, where they undergo further condensation with their telomeres becoming each attached to the nuclear envelope. So as we can see here, you have the paternal and the maternal finding themselves in the nucleus, and then they come closer to each other and begin the process of attachment. Now, these homologous chromosomes then get intimately attached, and as they get intimately attached, that moves them into another phase known as the zygoten, where as they become so intimately attached, they attach each other near the end of the telomere region, which is going to be referred to now as a synapses. Now, these synapses that are formed are important for genetic recombination, which contributes to the diversification of the gene pools that you find in the products or in the resulting cells. And at this point, the genetic information now would have been exchanged into those sister chromatids that are fused. So these attachments allow the process of crossover and then genetic information exchange, and these are stages or the point that is referred to as the chiasma or knots, which hold the chromosomes together. Now, once the chromosomes have been bound or intimately attached one to another, then they move into what is referred to as the pachyten stage. And at this stage, genetic recombination would have already been completed, and they form what is referred to as a synaptonemo complex, where they appear now as a single structure that is well joined together. So, having moved to the appearance of the synaptonemo complex, the chromosomes now will need to dissolve or to disjoin themselves at the synaptonemo complex, and that moves them to the diploten stage. Now, what is being dissolved here is not the chiasmata, but it is the separation of the synaptonemo complex, so that each of those homologous chromosomes now will appear and be on a stand alone. The genetic information has already been exchanged exchanged at this particular stage. And in females, you will understand also that there is going to be an arrest in the diploten until the onset of puberty, where the primary oocyte now will then complete the stage of meiosis 1 every month for waiting for the onset as well of the menstrual cycle. Now we have a simplified version of the process of gametogenesis, where I look at the process of gamete formation, and I would also encourage you to look out for that particular video and watch it so you understand the process of gametogenesis, and you will find that on the YouTube channel, the Brain Shakers Academy. And if you have not yet subscribed, remember to subscribe and hit the notification button so you do not miss any of the material that I will be posting onto that particular platform. Now, after the genetic material has already been exchanged and we have reached the diploten stage, it's now time for the chromosomes to reach a stage of diakinesis. Now, diakinesis is analogous to the pro-metaphase of mitosis, where the chromosomes will now begin to be aligned on the metaphase plate, and that takes us to the second phase of meiosis 1, which is the metaphase 1. So during metaphase 1, it is of short duration, and this is where the homologous pairs now are going to align themselves on the metaphase plate, and you already see the mitotic apparatus there appearing in readiness for the cell to get into the anaphase. So the stage of anaphase, this is where the homologs are at the pores. Now they will be pulled to the pores by the mitotic spindle fibers that are attached to their kinetochore. And we say the kinetochore is just a disc-shaped protein that you will find around the centromere, where the two chromosomes are attached in their middle part. That is where you find the kinetochore. Once the mitotic spindle fibers contract, they will be moving these homologous chromosome, one to this end and another one to the other. And now these homologous chromosomes, as they move to the pores, they still possess within them two sister chromatids that have not separated, because we are still under meiosis 1. And meiosis 1 is just the separation of the homologous chromosome. So this homologous chromosome part moves to one pore, and this other one also moves down to the other pore. And at this stage now, we would have separated the homologous chromosomes, awaiting now for the sister chromatids in this particular chromosome to then be separated once the cell enters meiosis 2. Now, that is anaphase, and it creates a space in between them so that the cell can then develop a furrow and be allowed to separate. So any mitotic spindle fiber that is not attached to chromosome will begin to elongate, moving the poles and the centrioles further apart and creating a larger space in between the homologous chromosomes, and the cell appears a little bit elongated, allowing for a furrow to form, and then there will be separation, and that takes the cell into another phase, which is going to be the telophase. Now, during telophase, you have the cell that is elongated and the furrow has already formed, and then it is just waiting to completely pinch when the cell gets into the cytokinesis. So within the telophase, similar to what happens in the stages of mitosis, what is going to happen now is that the mitotic apparatus which was there is going to be broken down into its building blocks, and it is no longer there, no longer appearing within the cell, and the nuclear membrane that had also disappeared comes back and reforms around those homologous chromosomes that have been formed, and you have your centrioles there that had moved with their asters to the pores, also surrounded and become the centrosome of that particular cell. Then the cell moves into the process of a cytokinesis 1, which just completes the process of dividing the two cells in one to another by completely pinching the cytoplasm in the middle and separates it into two daughter cells. Now let's look at what happens after the two daughter cells have been formed. So once the two daughter cells have been formed, the cell move from mitosis, from meiosis rather, move from meiosis 1 to meiosis 2. Now, without copying their DNA material, these two cells will also undergo a series of cell division in order for them to be able to produce each two cells. Okay, so they will undergo these similar processes, which is prophase, the metaphase, the anaphase, and the telophase. Now, these processes are similar to the processes that happen in the cell division known as mitosis. And you also notice that the process of meiosis 2 is where the sister chromatids now are going to be separated. Now, each of the sister chromatids will possess within them the genetic material that they obtained during the process of genetic recombination. So the resulting cells will still have the genetic information coming from the maternal side and from the paternal side, and the resulting cells will be 4 cells in total, because each of the 2 cells that have been produced will end up producing 2 daughter cells within them, and each of them will have a haploid number of chromosomes, which is just one set of chromosomes within them. So that is about the process of meiosis, which is basically meiosis 1 and meiosis 2, and the processes and stages that the cell undergoes are similar in both phases, with meiosis 1 being a little bit longer than meiosis 2 because of the prophase that contains 5 other sub phases. Now, if you like this particular video, I would like you to subscribe to my YouTube channel, which is the Brain Shakers Academy, and do not forget to give it a thumbs up and share. You might just be helping somebody else to understand the process of meiosis. And as always, it's a pleasure to have you, and thank you so much for watching, and I'll see you in the next one.